Welcome back, learners. So today we will be discussing another aspect of your microbiology, and that is very important in pharmaceutical industry, and also that presently in clinical microbiology, medical microbiology, the emergence of drug resistant bacteria and etc. So this particular lab is very important because it will tell you that how to determine your antibiotic or antimicrobial, or it may be antimicrobial susceptibility testing. So it is your antimicrobial susceptibility tests or testing. Okay, so this is your antimicrobial susceptibility testing. Now first and foremost question is that what is susceptibility? What is your susceptibility? Now susceptibility testing determines if bacteria is likely to be inhibited by any microbials. So suppose this is your bacteria, and this bacteria is likely to be inhibited by certain antimicrobials. So that is your susceptibility testing. Now susceptibility is based on certain parameters. So susceptibility is basically based on concentration, okay, required to reduce growth below the visible level, below the visible level. Okay, so that is your MIC, that is your minimum inhibitory concentration. So for that, you know that this antibiotic susceptibility testing has many classifications. So we will move on to your classification one by one. So this is your antibiotic susceptibility tests. So if I drag this to the other part of your, so this is your antibiotic sensitivity test. So antibiotic sensitivity test basically comprises of diffusion and dilution both. Now if it is diffusion, it is further classified as Kirby-Bauer method and Stokes method, and this method in totality is called as qualitative method. And of course we should also have a quantitative method to determine, that is your tube dilution and your agar dilution method, and diffusion and dilution is the E-test. So here we will be basically focusing, as we don't have all the different things in your syllabus, you only have Kirby-Bauer method in your syllabus, so I will basically first focus on your Kirby-Bauer method. So let's discuss about your Kirby-Bauer method. So Kirby-Bauer agar diffusion method provides a qualitative test, which I have already discussed. Okay. It is this particular results is interpretative of the following thing. That is, it tells us that whether a microorganism is your susceptible, intermediate resistant, mediate resistant, and your resistant bacterial isolates. So it will tell you that whether your bacterium is susceptible, intermediately resistant to the antibiotic, and you're resistant to the antibiotic. Now Kirby-Bauer antibiotic testing is a test which uses antibiotic impregnated disc. So you have your antibiotic AB impregnated disc. Okay, so you require this, okay, to test whether particular bacteria are susceptible to that particular specific antibiotic. And you know that A known quantity of bacteria are grown on the agar plate. So if I take an agar plate and you allow a confluence zone of the bacteria, onto that of your plate, and of course will be grown, and then you have to put this disc. Suppose this is your antibiotic disc, you put the disc on the plate. Okay, if the bacteria are susceptible to particular antibiotic, there is a zone of clearance all around the antibiotic. So you get to see a zone of clearance all around the antibiotic. So again, I will draw it again. So you will get to see a zone of clearance all around the antibiotic. So that is what is called as your zone of inhibition. So you get to see this is your zone of inhibition. Okay, so it is your zone of inhibition. So agar disc diffusion method has various parameters. So if we focus on those parameters, then let's discuss about those parameters. There is a storage condition. First is your antibiotic disc. So if you have antibiotic disc, so that storage temperature is very important, and the storage temperature is usually -20 degrees C minimum. -20 degrees C minimum. Then is your inoculum. How much inoculum you will be adding onto the particular plate. So that is your 10 to the power 8 bacteria per liter. Next is your incubation. How much time you will incubate. So that is your 35 degrees C. And of course, you will have ambient temperature. So this ambient temperature, I'm very sorry to say, it is the ambient air supply. So you have antibiotic disc, you know how much inoculum you have to add, you know the incubation temperature, that after addition of the inoculum, how much time you will take to incubate, and of course your ambient air has to be supplied. So let's move on to the procedure. So for procedure, I have two different images. Of course, again I will tell again and again in my video that I will always acknowledge all the image which I have taken from the Google. So I will acknowledge all those Google images which I have taken for preparation of my video. So I am very thankful for those images. So this is your antibiotic susceptibility testing. So this is one image which I will be discussing, and of course I will also add another image which will totally describe your procedures. Yes. So these are the two images which will basically best describe your procedure. So it will be best describing your procedures. So see these two images. So this will basically describe your procedure. So see, this is a Petri plate. So in step 1, you see the Petri plate. So this is your Petri plate. So what you have done is that you have a swab with you. Okay. So you have taken a 24-hour incubated culture. Okay. After you have taken a 24-hour incubated culture, what you do with this swab, you basically streak confluently throughout the plate such that you get a confluent growth after 24 hours of incubation. Now what you do, you start selecting, place your disc. So next important point is that, so first is inoculate the plate, second you place the disc. So you have disc impregnated with your antibiotics. Now you have to add this particular antibiotics, discs into the plate. So you have put the disc into the plate. Now after that you incubate for 18 to 24 hours. So 18 to 24 hours at 35 degrees C to produce your zone of inhibition. So in order to see that whether zone of inhibition is produced or not. If it is sensitive, it will of course produce zone of inhibition. If it is not, it will not produce any specific zone of inhibition. So this is further procedure. You can see it in details. The dispense antibiotic discs with the dispenser. So with the dispenser, they're dispensing the antibiotic discs into the inoculated agar plate and space antibiotic discs equidistant from each other and inoculate the plate with sterile forceps. So with the sterile forceps, see what they have done? They have inoculated the disc very, very carefully with equidistant placement of the disc. And then after they have gone through the incubation time period, they have got to see the zone of inhibition. And when they have got to see the zone of inhibition, with the help of your caliper or also a millimeter ruler you can use, they are finding out the zone of inhibition, the diameter of the zone of inhibition, to determine your antibiotic susceptibility. So this is basically the plate method by which you can do the thing. Now we will move on to the tube method. So let's take another image. Again, I will acknowledge the image, and this is your tube method. So for tube method, I am selecting an image to help you to describe the tube method of MIC, minimum inhibitory concentration. See what you have. If you get to see in this particular image, this will tell you the whole procedure. So this is your step 1. What is step 1? Step 1 is the tube dilution. That is volume of microbial rich broth. So they have taken the broth 120, 100, 80. So they have taken that, and volume of antimicrobial agent. So 0, 20, 40, 60, 80, 100, 120. Okay. So this they have been done, right? Then step 1, then is your step 2. Step 2 is incubate for 24 hours. And then comes your step 3 that determines the minimum inhibitory concentration based on the turbidity. So see that it is 0%, 16.7%, 33.3%, 50%, 66.7%, 83.3%, 100%. So as you see the turbidity, this is most turbid. Then the turbidity decreases with the increasing concentration of the antibiotic. A time comes when at a concentration you get to see that there is no growth. It is nearly transparent. It is not turbid. And then you have absolute turbidity. So this particular tube in which you get to see a considerable turbidity because of the antibiotic, then you can say that the microorganism is being inhibited or killed by the antibiotic, and that can be your minimum inhibitory concentration. So that is your tube dilution method. So next we will move on to the observation. So again, I will delete this and I will add another image for your observation. So next we will move on to the observation, and you know that in the observation, this is an observation. See, you will get to understand the importance of this particular test from this observation. You get to see 2 plates. So all are, see the confluent growth of the microorganism which is being done by swabbing, and they have put the disc. Here you see the zone of inhibition. It is less than 16 millimeter in diameter, and this particular zone of inhibition is more than 16 millimeter in diameter. So what does it mean? The Staphylococcus aureus is sensitive to the particular antibiotic, whereas Staphylococcus saprophyticus is resistant to that. It is novobiosyn. Okay. Now the question is that what is your standard? What is your standard MIC, and from where? From that, how will you compare it with that particular standard MIC? So for that, we have rules and regulation given by your CLSI guidelines, Central Laboratory Standard Institute, which will tell you that this are the standard MIC and how your MIC varies, and variation of the MIC will be ultimately interpreted as your sensitive, intermediate resistant, and your resistant microorganism. So from CLSI guidelines, you will be able to understand that after the test, whether your organism is sensitive, resistant, or intermediate resistant to the test antibiotic which you are basically using. So this is all about your antibiotic susceptibility test. So in antibiotic susceptibility test today, we have discussed the Kirby-Bauer method, and this disc diffusion method was there, that is your Kirby-Bowen method, and that was done in your agar plates. And also we have focused a bit of your antimicrobial susceptibility, that is your tube dilution method. So in the tube dilution